Each eukaryotic chromosome consists of a single, extremely long molecule of DNA. For all of this DNA to fit into the nucleus, tremendous packing and folding is required. The chromosomes are in elongated and relatively uncondensed state during the interphase of the cell cycle. The largest human chromosome contains about 0.3 billion base pairs. If it is in normal conformation, it would constitute a chromosome of greater than 10 centimeter long. How is it possible to fit all 46 chromosomes into a nucleus that is only 10 millimicrons in diameter? And at the same time, it has to maintain the DNA in the state that is accessible to enzymes and regulatory proteins. The answer lies in the remarkable manner in which the DNA molecule is packaged in eukaryotic cells. Nucleosome is a concept which explains or gives an answer to this question. Nucleosomes are the fundamental organizing unit of all eukaryotic genomes. Understanding how the proteins gain access to DNA binding site located within the nucleosome is an important for understanding the DNA processing including transcription, replication and repair. Nucleosome model is a scientific model which explains organization of DNA and associated proteins in the chromosome. Nucleosomes are the fundamental organizing unit of all eukaryotic genomes. It explains the exact mechanism of folding of DNA in the nucleus. The nucleosome is the lowest level of chromosome organization. DNA and histones are organized into repeating subunits called nucleosomes, and each nucleosome contains about 146 base pairs of DNA wrapped almost twice around the disc-shaped complex and 8 histone molecules that is also called a histone core. The model was proposed by Roger Kornberg in 1974 and is the most accepted model of chromatin organization. Now, what do you mean by DNA packaging? The arrangement of DNA on chromosome through nucleosome assembly is known as DNA packaging. Wrapping of DNA molecule around the histone protein is known as nucleosome. The DNA folded 10 to the power of 4 times to its original length and become a metaphase chromosome. The folding of DNA is started when proteins called histones interact with DNA. The nucleosomes are the fundamental packing unit particles of the chromatin and give chromatin a beads on a string appearance in an electron micrograph as you see in this photograph. What are the components of a nucleosome? It contains H1, H2A, H2B, H3 and H4, which are the 5 major types of histone proteins involved in eukaryotic DNA arrangement, in which H2A, H2B, H3 and H4 creates the nucleosome assembly. Each nucleosome is made up of 8 histone molecules, each H2A, H2B, H3, H4 two times in a single nucleosome. The DNA wraps around it to form the next level of organization. In chromatin, the DNA extends as a continuous thread from nucleosome to nucleosome. Each nucleosome bead is separated from the next by a region of linker DNA, which is generally 54 base pairs long and contains single H1 histone protein molecule. Generally, DNA makes two complete turns around the histone octamer, and these two turns, that is 200 base pair long, are sealed off by H1 molecules. The histone core of each nucleosome consists of two copies of H2A, H2B, H3 and H4 proteins. The remaining histone H1 resides outside the nucleosome core. Together, the core particle is associated H1 histone and is called as chromatosome. And each chromatosome encompasses about 167 base pairs of DNA. Chromatosomes are located at regular intervals along the DNA molecule and are separated from one another by linker DNA of 30 to 40 base pairs. The linker DNA is the piece of DNA that connects adjacent chromatosomes. Each of the histone proteins that make up the nucleosome core particle has a flexible tail containing from 11 to 37 amino acids that extend out from the nucleosome. Now, how the DNA histones are tightly associated with the histone proteins. The positively charged amino acids in the tails of the histones interact with the negative charges of phosphates on the DNA and keep the phosphates on the DNA, keeping the DNA and histones tightly associated. Higher levels of chromatin structure, that is organization. A DNA molecule is the lowest level of chromatin organization. The first level is the formation of 30 nm fiber, and these fibers form a large coiled loop held together by non-histone proteins like actin, alpha and beta tubulin, myosin, etc. And these are called as scaffolding proteins to form extended chromatin, which is 300 nm in diameter. As far as 30 nm fiber is concerned, two different models have been proposed. One is called as the solenoid model and another one helix model. In solenoid model, in which a linear array of chromosomes are coiled, whereas in the helix model, the nucleosomes are arranged in a zigzag ribbon that twists or supercoils. So most of the evidences suggest that helix model is the ideal one. The next higher level of chromatin structure is the formation of 300 nm fibers. At the next level, the 300 nm fibers along with the protein of a network of proteins leads to the formation of 1400 nm structure, that is a highly condensed chromatin structure, that is called as a chromosome. This figure illustrates the organization of DNA within the chromatin structure. The lowest level of organization is the nucleosome, in which a DNA strand is wound around outside of a histone octamer. Beads on string an extended configuration of nucleosomes formed by the association of DNA with our four types of histones. Nucleosomes are connected to one another by a short stretcher of linker DNA. At the next level of organization, the string of nucleosomes is folded into fibers about 30 nm in diameter, and thus fibers are then further folded into higher order structures, that is 300 nm fibers, and further into 700 nm fibers, and later on 1400 nm structures, that is the metaphase chromosomes. The replicated chromosome, that is the two chromatids, from a dividing cell with all DNA of the chromosome in the is a very highly compacted heterochromatin.